Okay, chapter 5, viral structure and life cycles. So, previously we kind of mentioned viruses, and I mentioned that it's kind of in debate whether they're living things or they're not. But when it comes to microbiology, they are too small to be seen by the naked eye, and they can be pathogenic, they can be infectious, they can make people sick. So, it's still part of micro. So, an introduction to viruses. Viruses infect every type of cell that there is. They can infect bacteria, algae, fungi, protozoans, plants, and animals. There's nothing on the planet that can't get infected by some type of virus. Now, viruses may not transfer between those things, but it can still viruses still exist that can infect everything. Seawater can contain up to 1 million viruses per milliliter. So per 10 drops of water, there can be up to 100 million viruses in there. For many years, the cause of viral infections was unknown. Um, you got to remember that even when we had microscopes, you can't see a virus through a microscope. So um, they thought something was in there, but they couldn't really prove it at the time. So Louis Pasteur postulated that a living thing smaller than a bacteria caused these diseases that they couldn't find bacteria for. He also proposed the term virus, which is Latin for poison. Um, Ivanovsky and Bergernik showed that a disease in tobacco was caused by viruses. They were the first ones that actually had empirical proof. They could prove that there was something there. Um, Loeffler and Forsch discovered an animal virus that caused foot and mouth disease in cattle. Um, they were able to prove the existence by filtering the virus out. Okay. These early researchers found that when fluids from a host organism, a sick organism, pass through porcelain filters designed to trap bacteria, the filtrate remained infectious. In other words, whatever it was so small, it went through the filter and was able to infect whoever got exposed to it. This result proved that cell free fluid, in other words, I filtered it, there are no cells in there, contained some type of agent that could still cause an infection. It's kind of an indirect way because I'm still not showing what this is. I'm still not able to isolate it and go, here, look, this is what it is. But I am able to make sure that there's no cellular material, and yet the next organism that I expose this to still gets sick, meaning there has to be something in that fluid that can be transferred, that can be infectious, that can be pathogenic. Okay? So, this is what I was talking about. Viruses, are they alive or what? There are two sides to the debate. Since viruses are unable to replicate independently, literally, if I put a virus on the table and leave it there, it's not going to do anything, from a host cell, they are not living things and should be called infectious molecules because by themselves, they can't do anything. They don't have an independent life. Okay? But the other side of the debate is even though viruses do not exhibit most of the life processes of cells, they can direct them, they can change them. Thus, they're certainly more than inert or lifeless molecules. The fact that they can infect, take over the machinery of a cell, and make more copies of themselves, this is the argument for they're living things. But the fact that they can't live independently is the argument that they're not living things. Because living things can live by themselves. Viruses cannot. They cannot reproduce. They don't have metabolism. They don't have any of that. So, viruses, if we're going to describe them, can be better described as being either active or inactive rather than alive or dead. Active means that they're multiplying, they're in something, and they're creating issues. Inactive means that they're not inside something and they're not active at that point. They're basically dead. Okay? So, um, these are, hang on one second. Okay, so important properties for viruses. These are all key characteristics that are going to set viruses apart from the other organisms that we've discussed as far as microbiology. Okay, this is table 5.1, so it's in your book. Um, they're obligate intracellular parasites of bacteria, protozoa, fungi, algae, plants, and animals. So intra means inside, cellular. So they have to be inside of a cell in order for them to function. They're obligate. They're obligated to be inside a cell. They're ubiquitous in nature and have had major impact on the development of biological life. Ubiquitous means they're everywhere. So, as I said, they infect everything. The fact that they do that, some of them can cause mutation, and that actually changes the organism that they have infected. Um, they're ultra, ultra microscopic in size, ranging from 20 nanometers to 450 nanometers in diameter. They're really, really small. You're not going to see them in a microscope. You will be able to see them using an electron microscope, but not just a light microscope or compound microscope like we use in the lab. Okay. They're not cells. Their structure is very compact and very economical. You have genetic material, you've got a capsule or a capsid on top. Basically, it's a room holding genetic material. That's it. It would be almost like a nucleus all by itself, except nucleus also has other stuff inside. These don't. Um, let's see. They do not independently fulfill the characteristics of life. So, for example, one of the characteristics of life is reproduction. If I'm not inside of another cell, I can't reproduce, I can't make copies. They're inactive macromolecules outside of the host cell and active only inside the host. Again, it's a good way to kind of describe them as active versus inactive or dormant, because if they're not inside a cell, they can't do anything. They just sit there. Um, Basic structure consists of a protein shell, a capsid, surrounding a nucleic acid core, the genetic material. Nucleic acids can be, this is also something that's very unique. They can be double stranded DNA, which is what we have. They can be single stranded DNA, nothing like we have. They can be single stranded RNA, again, not really what we've got going for us. Or they can be double stranded RNA. Even bacteria don't use RNA as genetic material. So that's very unique. Molecules on the virus surface impart high specificity for attachment to the host cell. Viruses have basically what you would kind of describe as a key that fits into a very specific lock. So you've probably heard about these because of COVID. They're called spike proteins. Spike proteins are literally these proteins that stick out of the surface of the capsid that fit only into the right place. So this key, the spike for COVID, fits humans' um, protein on their cell surface. And to get even more specific, it has to be in the right place. So the cells of our skin don't have this lock for the key to fit into. The only place it really works is in our lungs, because our lungs have the right lock for this key. So when we talk about specificity for attachment, that's what we mean. COVID is not going to go and infect a turtle. Human COVID isn't going to go and infect a turtle. It's like HIV. HIV can't go and infect a dog. It's human immunodeficiency virus. So it's specific to humans. They multiply by taking control of the host's genetic material and regulating the synthesis and assembly of new viruses. Again, I, as a virus, invade your cell and turn it into a virus factory to make copies of myself. I can't do it by myself, but I can take over your cell and I can make your cell make copies of me. They lack enzymes for most metabolic processes. So things like lysozyme and amylase and things like that, enzymes that are in our cells, they don't have them. They don't have the capacity to make them. They don't have ATPase to make ATP. The things that you would associate with a living cell aren't there. And they lack any type of machinery for making proteins. That capsid that is covering up the nucleic acid material in the middle is a type of protein. They have no way to make that. All of the machinery, the ribosomes, the endoplasmic reticulum, the uh, Golgi to finalize, they don't have any of that. So the machinery that you would um, link with or kind of put together with making a protein, you've got the capsid, you've got the genetic material, you've got the spikes on the outside, the end. All of the other stuff, the capsid, the spike proteins, have to be made by the cell that they have invaded. So when I say I make it into a virus factory, I'm talking about all those parts and pieces are getting made by the cell, not the virus. Okay? So, how small are viruses? Um, they are the smallest infectious agents. Uh, some of the largest viruses are about as large as some of the smallest bacteria. Sorry, guys. Okay, this is a yeast cell. Okay, really, really large. That's E. coli, that's Streptococcus. So, the Mimi virus is similar in size to Rickettsia. Rickettsia is zero point 0.3 micrometers. It isn't even 1 micrometer large. A micrometer is 1,000 times smaller than a millimeter. There we go. Mimi virus, which is basically the biggest virus, is about the size of Rickettsia. As we go down from there, you'll notice we've got herpes, rabies, HIV, influenza, adenovirus, bacteriophage, which infects bacteria, polio, yellow fever. This is a hemoglobin molecule, part of what holds oxygen inside of your cells. Yellow fever and hemoglobin aren't that different in size. So when we say they are submicroscopic, they're teeny, teeny, tiny. They're teeny, teeny, tiny. Okay, so remember I said they're pretty simple as far as structure. Viruses don't have any type of cellular structure found in either prokaryotic or eukaryotic cells. Fully formed, a virus is able to establish an infection in a host. We call that a virion. There are two major parts to a virus: the capsid, which is the protein covering for the nucleic acids, okay? And then the nucleic acids that are inside of that pocket of capsid. It's found in the central core. And again, it can be DNA or it can be RNA. So we've got the covering and we've got the core. The covering is going to be the capsid. Some viruses have an envelope, not all of them. And then inside of that, you've got the nucleic acid molecules, either DNA or RNA. And again, some viruses, not all. There may be some matrix proteins, but again, some, not all. All right, so the viral structure, the outer covering. So let's talk about the capsid. It's a protein covering that protects the nucleic acid inside. That nucleic acid is the instruction manual. It is very important that we protect that, okay? So we've got identical subunits called capsimeres. If you look at this shape here, it is a bunch of triangles that have been put together. Each one of those triangles is an exact copy, but they can be put together kind of like, I don't know, red Legos, and you can make something out of that identical subunit, okay? There are two major types of capsids. You've got helical and you've got icosahedral. There we go. Okay. Now, I said some have an envelope, some don't. So, in this picture, we've got an envelope, okay? It's an optional outer layer covering to the capsid because you can still see the capsid's in here. It's embedded inside of this envelope. Now, this is kind of the interesting thing about the envelope. Usually the envelope is made from part of the membrane of the cell that was infected. So this is actually the infected cell's membrane. It's the outer covering of the cell that I went in and turned into a virus factory. Regular host membrane proteins will be replaced by the viral proteins. Remember I talked about the spikes? Okay. So let's actually talk about the spikes. If you look here, we have these projections coming out of the capsid or the envelope, okay, these little thingies that are sticking out that dock with the host cell. So any of you who have ever seen any type of sci-fi show, when you dock, you have to have the right kind of connection so that you can move between ships, right? So the spikes are what give viruses their specificity. When it comes to viruses, they have to be in the right host. So if I get the flu and I'm coughing around my cats or dogs, you'll notice that they never get sick. One of the reasons is because they don't have the right lock for the spike protein to fit into. As well, with the flu, it has to be in the right space in your body to actually make you sick. Somebody with the flu can come up and lick my arm and I will not get the flu because the virus needs to be in your lungs. Okay? That's why that spreads it into the air, which means that when you go, it goes into your lungs, and now I've got the perfect environment to infect. All right. So, what's at the core? Viruses will either contain RNA or DNA, but not a combination of the two. So it's one choice or the other, and that's the only options you get. So RNA or DNA. The number of viral genes is actually pretty small compared to that of a cell. They really only possess the genes needed to invade the host and redirect their activity. That's what it's going to do. Okay? Viruses exhibit a wide variety of configurations of DNA and RNA. Like I said, it can be DNA that's single-stranded, it can be double-stranded, it can be circular DNA, it can be linear DNA. It kind of just depends on the virus. Same with the RNA viruses. It can be double-stranded, but more often it's single-stranded. It can be positive sense RNA or negative sense. Basically, it can be positive sense, meaning it's ready to be read and made into a protein, or negative sense means that it has to be converted into the right orientation before you can translate this piece of RNA into a protein. Now, retroviruses. HIV is an example of a retrovirus. They actually carry their own enzymes to create DNA from their RNA. So, one of the things that's kind of evil about HIV is it invades, converts its RNA to DNA, and then will insert that piece of DNA into your cells. And it just sits there. Your cell doesn't know that it's infected because it's not making viruses, but it just sits there and waits. And eventually, some event activates it and it'll pull it out and start making HIV. But with retroviruses, we start with RNA, we convert it to DNA, and then the DNA is what we actually use. Okay. So, different types of viral nucleic acids. You do need to be kind of familiar with at least these 7 examples. So, an example of double-stranded DNA would be variola virus. This is what causes smallpox. Herpes simplex 2 would also be an example. This is what causes genital herpes. Single-stranded DNA, an example of that would be parvovirus. Um, Erythema infectiosum, basically it's going to cause red skin. Okay. Um, an example of an RNA virus that is positive polarity, okay, is poliovirus, causes poliomyelitis. An example of a single-stranded RNA virus that is negative polarity, it's backwards. That's going to be influenza, which causes influenza, okay, influenza virus. An example of double-stranded RNA would be rotavirus. Rotavirus causes gastroenteritis, it causes intestinal issues. A single-stranded RNA reverse transcriptase virus, in other words, it's a retrovirus, is going to be HIV. It causes AIDS. All right.